Hello everyone. Just a couple of bookkeeping things. This is a bit of a review. We actually talked about this before the break, but since it's been so long and since this is such an important topic, I'd figured I'd put a review up about it. I'm going to do these online lectures in a little bit of a shorter format. So rather than one long one, an hour or longer for each one, I'm going to do shorter ones, maybe 20 minutes, 15, 20 minutes, and we'll do 2 or 3 for each of the long hour lectures that we would have in class. And then that way you can review them and go over them. So this is the last part of 2.5, which we talked about right before the break. But it's important enough I want to do it again. If you're comfortable with gene duplication and you feel you know that already, then you may want to just skip over this and go straight into the next one, which would be 2.6. I've also updated the SLOs so they reflect and will reflect those shorter lectures and segments, so you can just kind of use those as a guideline as you're taking notes and reviewing. Okay, so again, as organisms evolved, got more and more complex, their genomes got larger also, and this happened via mutation events. And there are basically 2 ways that you can make a gene. One, you could make it from scratch, which we'd call de novo, or you can make it from a copy of something that's already there, either a whole gene that's copied again, so you have 2 of them, or in some cases you could do an unequal crossing over, which would result in extra genes. That's really just another way to copy genes, because really if you just do recombination, you're just mixing and matching what you already have. You don't get anything new. So really it's either de novo, mutation, or gene duplication. And gene duplication is by far the most common way that we get new genes. In fact, probably 99% of all genes can be put into gene families. Maybe not quite 99%, but a large majority could be put into gene families, and we can recognize a paralog in the genome. Okay, so if there's a slipping error during DNA replication, if this happened to a large enough region of DNA, then we could copy an entire gene. But more commonly, probably we get other mutation events, like an unequal crossing over event during meiosis, which swaps entire chunks of chromosomes. So some gametes end up with extra genes and other gametes are missing genes. So if that gamete with extra genes then goes on to fertilize or be fertilized, then that organism is now carrying extra genes. That becomes part of their genome. And it's like any mutation. Many times it'll be a bad thing. Those organisms will die or won't pass on their genes at very high frequencies. And eventually those duplicated genes will die out, won't become part of that species genome. But if it's effectively neutral, or if it provides an advantage, which mutations can happen, which can happen to mutations sometimes, then it's going to be selected for. And so this is just showing that we end up with extra copies of a single gene, or maybe even entire clusters of genes that are duplicated. Okay, so just be aware that it's a very common way that we get new genes. And this occurs for all genes, not just homeotic and developmental genes, but it's especially important in the developmental genes. So here is hemoglobin, right, which is not a developmental gene. It's a tissue-specific gene. It has a very critical function in most organisms, but there's really no reason why Daphnia would need these 8 different copies. And so the idea is there might have been multiple, maybe an initial duplication from an ancient hemoglobin gene, and then another one to get to 4, and then another one of the entire cluster to get to 8. And so we end up with probably a lot of redundancy here. That gives you backup copies and also some flexibility and movement in the future. One of them could mutate, or many of them in this case could mutate, and you'd still have all the original ancestral function because of the other original genes. Okay, so once a duplication event occurs, these slippage events are not any more likely, but unequal crossing over is, because when chromosomes line up during meiosis, they do so based on their genetic similarity, and basically matches in different homologous chromosomes find one another, and so they line up evenly most of the time. But if we have the same gene duplicated many times, here they're kind of color-coded, notice that we can get a mismatch. And so if crossing over occurs there, we end up with one gamete with extra genes, and even one, since this crossing over event occurred right in the middle of a gene, one that's kind of a Franken gene here, but they still have the original 3 genes, the orange, the purple, the blue. And then of course this gamete is missing material, so that would be a deletion mutation there in that gamete. and a duplication with a little bit of recombination occurring also. So really, if crossing over occurs within a gene, you could get not only a gamete with duplicated material, but also with some recombination. So you've got a new flavor of gene that's maybe allele A and allele B are mixed together to make a brand new combination there. That might do something a little bit different. Okay, but again, it's a mutation, maybe good, maybe bad. If it's good, it's going to be kept. And now that organism has extra genetic material and the extra ability to do new, unique things. Okay, this has been a very important mechanism and force in the diversification of developmental genes, particularly the Hox genes. So here are Hox genes and members of the homeodomain family that no longer have canonical Hox function, but are still part of that family and resulted from an ancestral duplication event. And so you need to be very, very clear about these ideas of gene families, which are just groups of duplicated genes. And so within them, and looking across species, we can define both paralogs and orthologs. Okay, review that. Both of them are types of homology. A paralog is a homologous gene that can be traced back to a gene duplication event. Okay, and so labial from a Drosophila and Proboscipedia from a Drosophila would be paralogs. They would eventually have an ancestor here, but that's a duplication event. Whereas if I took labial from a Drosophila and labial from a beetle, those would be orthologs because they would go back to a speciation event. So in this diagram, we've identified different members of the gene family. Everything that's color-coded with these little round highlighting areas are paralogs. All the caudal genes in different species are paralogs of one another. Okay, so just be aware of that, that there is some interesting patterns that we can identify once we've looked at these complex gene families. And so by doing gene phylogenies rather than species phylogenies, we can identify gene duplication events, right? So those are paralogous copies that trace their ancestry back to these duplication events. And then we can identify these clusters of speciation events. So humans are more closely related than mice, they're both mammals. We see that same pattern over here again. And so we have a duplication event from a single ancestral engrailed gene to two, and then another duplication event in this lineage, another one in this one lineage. So zebrafish now have four engrailed genes. Okay, so just be aware, you might see something a little simpler than this, but you should recognize that if you have the same species represented twice on a phylogeny, that some of these are going to be gene duplication events. Okay, and so here it is on the species phylogeny where we have mapped those gene duplication events onto that phylogeny. So again, a very important critical mechanism for getting extra genetic diversity that allows you to do more things, increases the potential phenotypic diversity of species. Okay, so Hox complexes, we've got individual duplications. We also have entire complex duplications across the vertebrates. So it's very, very important. Okay, so once a gene is duplicated, that immediately gives redundancy. Right? And because there is redundancy, it's very likely, in fact, probably by far the most common outcome of a duplicated gene is that you're going to lose one of the copies. By loss, we just mean that maybe the DNA is still there, but it's no longer a gene. It's no longer transcribed. It's no longer translated. And so for a period of time, if we can still recognize a duplicated copy of a gene as part of that gene family, but if it's no longer being transcribed and translated, we call it a pseudogene. Okay, and so here we have, this is not a developmental gene, but an important tissue-specific gene, globin, and we can have functional paralogous copies, right? So the gamma and the alpha here are paralogous copies, and then we have these genes that are marked here with this little Greek symbol that are pseudogenes. They're no longer functional, but they still have enough sequence similarity that we can recognize them as a duplicated version of a once functional gene. Okay. Now, the other outcome is new function for the genes. And so we're going to have divergence here. And in fact, let's do this. I'm going to add a little bit of an icon here. So I'm going to insert, just to show you what we mean. This slide's not the best. I took it from another publication. But if we insert a shape here, and let's just put I don't know, square here to represent that there's a mutation that occurs in the gene itself. And so now when I look at this, I've got a different gene. Notice the regulation is identical, but the gene is different, right? And so this means that we could be turning on both of these target genes in the same tissues because they're regulated the same, but now we have slightly different products. So diversity in gene expression. So the technical term for new function is neo-functionalization, just a fancy term. But in addition to neo-functionalization, let's skip this part, it's a little bit more in depth than we need to get into. In addition to neo-functionalization, we could also have brand new regulation. So in this one, the genes are identical, right? Same exact gene, but it's turned on now in different circumstances. And so this shows how we end up with pleiotropy. So in fact, let's put that here. New regulation results in pleiotropy. Sorry, I can't type. There we go. We'll make that just a little bit smaller so you can get it all on there. Okay? So this is a very, very common thing. And so if you have a gene that is still identical but used in two different tissues, that may be because the gene itself has not changed. It's maybe a very important gene. And so even after it duplicated, it didn't change, but we changed the regulatory function. So we have two identical copies, but this one might be turned on in the brain. This one might be turned on in, say, developing muscle tissue. So that same gene can have separate independent functions. And then later they may diverge, but very commonly they'll be very, very similar and conserved. Okay? So review that. Be sure that you're very, very familiar with gene duplication. It's a critical thing. You're going to see several questions, probably even some essay questions on the exam, which is coming.